Hello students, today let us discuss about the organs of the immune system. Introduction. The immune system of our body consists of complex network of morphologically and functionally diverse organs and tissues called lymphoid organs. The lymphoid organs are functionally classified into groups: primary and secondary lymphoid organs and tertiary lymphoid organs. They provide the sites for the growth, development, and maturation of lymphocytes. They are joined together by blood vessels and lymphatic system of the body into a functional whole during an immune response against antigen by carrying the lymphocytes to and fro in different areas of the body. Primary lymphoid organs. The primary lymphoid organs provide microenvironments for the development and maturation of the lymphocytes. Thymus and the bone marrow are the primary or central lymphoid organs. Immature lymphocytes mature and becomes an immunocompetent cells capable of mounting an immune response in the primary lymphoid organs. Bone marrow. Bone marrow is the site of B cell origin and development in most mammals, including humans and mice, but in the brucha of Fabricius in the birds. Immature B cells proliferate and differentiate into mature B cells within the bone marrow after arising from lymphoid progenitor during hematopoiesis. The bone marrow contains a group of cells called stromal cells, which supports the growth and maturation of B cells. The stromal cells constitutes the fat cells, the endothelial cells, the fibroblasts, and the macrophages. B cells with self-reactive antibody receptors are eliminated within the bone marrow. Thymus. Thymus is the first organ to produce lymphocytes and sites for T cell development and maturation. It is a flat, bilobed organ situated above the heart. The lobe of the thymus is separated by trabeculae into lobules. Each lobule has two compartments, the cortex and medulla. The outer cortex is densely packed with immature T cells called thymocytes, whereas the inner medulla is sparsely populated. The thymocytes are surrounded by epithelial cells, also called nerve cells, macrophages, and dendritic cells that influence the development of thymocytes. The thymocytes undergo thymic selection, removal of T cells that express receptors for self molecules during maturation. Therefore, most of the T cells produced in the thymus die here. Thymocytes mature to functional T cells in the cortex, then migrate to the medulla where they encounter dendritic cells and macrophages, then pass into the peripheral blood circulation. Mature T cells expresses either CD4 or CD8 molecules are transported to the secondary lymphoid organs. Before we move into the secondary lymphoid organs, I would like to discuss the lymphatic system. There are two circulatory systems in the body, the blood and the lymph. As blood circulates under pressure, components of the blood leak through the thin wall of the capillaries into the surrounding tissues and are known as interstitial fluid. Most of the interstitial fluid returns into the blood through the capillary membranes. The remaining interstitial fluid, called lymph, flow from the connective tissue into the lymphatic vessels where lymph nodes are found. The lymph carries antigens from the tissue to the lymph nodes where immune responses are initiated. Lymphocytes enter the lymphatic circulation directly from the blood, to which they return via the thyrocyte duct, the largest lymphatic vessels draining into the circulation close to the heart. Several afferent lymphatics brings the lymph to a particular node and a single afferent lymphatics vessel carry it away. Secondary lymphoid organs. The secondary lymphoid organs are those organs that can trap antigens from defined tissues and are the sites where mature lymphocytes encounter antigens effectively. The lymph nodes and spleen are the secondary lymphoid organs. Lymph nodes. Lymph nodes are encapsulated bean-shaped structure packed with lymphocytes, macrophages, and dendritic cells. Morphologically, a lymph node can be divided into three concentric regions: the cortex, the paracortex, and the medulla. The cortex is the outermost layer packed with B lymphocytes, macrophages, and follicular dendritic cells, and contains primary lymphoid follicles. After antigen activation, the primary follicles enlarge into secondary follicles with germinal centers packed with proliferating B lymphocytes and plasma cells, interspersed with macrophages and dendritic cells. Germinal centers are a site of B cell activation and differentiation into plasma cells and memory cells. Just beneath the cortex, there is paracortex, which contains T lymphocytes and some interdigitating dendritic cells. The antigens present in the paracortex are trapped by the dendritic cells, which present the antigen fragments to T cells. Medulla is the innermost layer of a lymph node, sparsely populated with antibody-secreting plasma cells, travel from the cortex via lymphatic vessels and has high concentration of antibody. The lymph nodes efficiently trap antigens that enter via the afferent lymphatic vessel and empties the lymph into the subcapsular sinus through a single efferent lymphatic vessel. Lymph nodes filter foreign antigens with the help of macrophages, T cells and B cells, which bring about immune response by generating antibodies and antigen-specific T cells. Spleen. Spleen is the largest secondary lymphoid organ with ovoid structure situated high in the left abdominal cavity. The interior of the spleen is compartmentalized into structures separated by trabeculae. The compartments are red pulp and white pulp. The red pulp consists of the erythrocyte intermingled with many macrophages and dendritic cells, few lymphocytes and plasma cells, where all and defective red blood cells or RBCs are destroyed and removed. Whereas the white pulp surrounds the splenic artery, forming a periarteriolar lymphoid sheath, also called pulse, rich in T lymphocytes. The marginal zone of pulse is populated with lymphocytes and macrophages, which trap blood-borne antigens and present it with class II MHC molecule to T helper cells. Then, the activated T helper cells further activate B cells. Approximately 50% of B cells, 30 to 40% of T cells constitute the spleen cells. Tertiary lymphoid tissues. The tertiary lymphoid tissues are those tissues which normally possess fewer lymphoid cells than secondary lymphoid organs. This tissue play an important role during an immune response by undergoing a rapid and substantial increase of the lymphoid cells. Most prominent of these are various mucosa-associated lymphoid tissue or MAL, cutaneous associated lymphoid tissue or CAL, and the intraepithelial lymphocytes or IEL, mucosal associated lymphoid tissue or MAL. The mucous membranes lining the digestive, respiratory and urinogenital system are prone to attack by pathogens. These linings are defended by a cluster of non-encapsulated organized lymphoid tissues known as mucosal associated lymphoid tissues or also called MAL. There are several types of MAL: gut associated lymphoid tissue or GALT and bronchus associated lymphoid tissue or BAL are the two major types of MAL. GAL include the tonsil, the appendix, and the Peyer's patches in the intestine. BAL is structurally similar to GAL and consists of large collections of lymphocytes. It is found primarily along the main bronchus in the lungs. Cutaneous associated lymphoid tissue or CALT. The skin is an important barrier to the external environment. The outer epidermal cells of the skin secretes a number of cytokines and induce to express class II MHC molecules to functions as antigen presenting cells or APCs. The interdigitating dendritic cell of the epidermis internalize the antigen by phagocytosis or endocytosis, and they function as potent activator of naive T helper cells by presenting antigens with class II MHC molecules. Intraepithelial lymphocytes or IELs. The outer mucosal epithelial layer contains intraepithelial lymphocytes. Many of these lymphocytes are T cells that express unusual receptors, gamma delta T cells receptors or gamma delta TCRs, which exhibit limited diversity for antigens. CD4 positive and CD8 positive T cells as well as macrophages are also present in the dermal layer. Most of dermal T cells were either previously activated cells or are memory cells. They are well situated to encounter antigens that enter through the intestinal epithelium. In conclusion, I would like to state this few sentences. Most of the immune response to specific antigens are precipitated in the lymphoid organs. The lymphoid organs are classified as primary, secondary and tertiary. Primary lymphoid organs include the thymus and the bone marrow. It provides sites for maturation of lymphocytes and become antigenetically committed cells. The lymphatic system of the body collects fluid that accumulate in the tissue and return the fluid to the circulation via the left subclavian vein. It also delivers antigens to the lymph nodes. Lymph nodes trap antigens from lymph, whereas spleen traps blood-borne antigens. Tertiary lymphoid tissues, including mucosa-associated lymphoid tissue, cutaneous associated lymphoid tissue, and intraepidermal lymphocytes, interact with antigens that enter the body from the gastrointestinal, respiratory, and urinogenital tracts.